a lot of energy money to make a protein. So in some of their growth stages, bacteria actually spend like 50% of their energy expenditure is actually being spent in making protein. And this energy required for the protein making, it starts before you even get to the ribosome, which is the protein making machinery. You actually have to activate the amino acids and charge the tRNAs, so that you're paying upfront this big cost, so that the ribosome, this will actually convince the ribosome that it wants to use the amino acid. And by putting in this energy expenditure, we can make sure that this kind of helps pay for the fidelity to make sure that we're adding the right amino acid. And so the tRNA synthetases, these aminoacyl-tRNA synthetases, these aaRS's, they're going to be responsible for doing this amino acid activation, so attaching the AMP part of ATP onto the amino acid, and then for transferring that to the correct transfer RNA. And then later we'll have some energy making happen at the ribosome, making sure that the right tRNA is brought, and then that it gets pushed out of the way once you formed a peptide bond. But it's actually this pre-charging is actually, there's proofreading that goes into it, because when the ribosome gets an amino acid, gets a tRNA, it's just going to recognize it by its anticodon. It's not going to recognize the amino acid that's actually on it. So in order to make sure that we add the right amino acid, we have to have this aaRS be able to recognize the right amino acid and the right tRNA, put them two together, and now it's off to the ribosome. And so how it does this is some really cool stuff about amino acid tRNA synthetases, and it actually, so here we're putting in an energy expenditure of 1 ATP, but it's like the equivalent of 2 kind of, because it is you're making AMP instead of ADP, so we're taking off 2 phosphate groups instead of just 1. So each of these phosphate bonds is like a high energy bond. It's a high chemical energy, potential energy. Basically, these bonds, it takes a lot of energy to make them, but you get that energy back when you break them. And so this energy getting back is gonna be what we're using to help make things happen. So make this transfer happen, make the ribosome move over when it's that sort of thing. And so because of that, you then have to spend an ATP in order to regenerate the ADP, which then you can use the ADP with that cool ATP pump thing, where you're making ATP like you normally do, like in the mitochondria. But first you have to make ADP, so it's like you spent 2. And then when you get to the ribosome, and you have the factor that's going to bring it in, that's going to spend a GTP. Then the elongation factor, that's going to spend another GTP. So you have 1 ATP, 2 GTPs, but kind of like the equivalent of having 2 ATPs because of that other one. So a lot of energy, but it's important because we want to make sure that our proteins are made correctly. So how do these amino acid transferases work? Let's go in. So you probably noticed by the intro that this is going to be a bit more of a detailed talk, less of the background. And so if you want more background, I have lots of things on translation and that sort of thing. But for now, just we're going to assume that you have that background. So we're going to be talking about this process of translation, as well as what we have to do before translation, and really focusing on that before part for the most part. So translation is this process where we're making a protein by linking together the protein letters. So by linking together the amino acids through these peptide bonds. And this is going to be facilitated by this complex of proteins and proteins and RNA called the ribosome. And so the ribosome looks something like this. It has these 3 sites, and what it's going to do is it's going to actually read the instructions that were in the messenger RNA and piece together the amino acids that the messenger RNA is dictating. And the way it's going to do this is it's going to have the corresponding amino acids brought to it by a transfer RNA, by a tRNA. So these transfer RNAs on one of their ends, they have the amino acid that matches the one to be added. And then on the other end, they have this anticodon. So basically, a codon is one of these 3-letter RNA words. So 3 RNA letters is going to be a codon, and it's going to spell one amino acid. And so the tRNA that matches, so it matches in the sense of it's complementary. So if there's an A, there's a U. If there's a U, there's an A. If there's a C, there's a G. Then it can come and bind. And when it binds, then it's bringing the complementary amino acid. Just a little technical note is that because you actually have some tRNAs will actually recognize multiple codons. So there's this third position. So the first 2 positions are going to be these like strict ones, but then the third position is called the wobble position. And so sometimes you have a tRNA that is going to have the same amino acid, and so you can basically use the same tRNA for multiple for recognizing multiple codons in some instances where you have, so the first 2 would match, but then you would have a different wobble position. But the technical note aside, what's going to happen is that you're going to have an initiation step, and then the things are going to get started. So in your ribosome, you have these 3 sites, E, P, and A. The P site is where you're going to have the growing peptide chain, so the nascent chain. And then the tRNA is going to come in with the corresponding amino acid. And then the ribosome is going to help facilitate the passing of the chain to the incoming tRNA. So this chain is going to get passed from this this peptide or this tRNA that's in the P site. It's going to get passed on to the A site, so to the newly arrived one. And then things are going to get shifted over. And this is going to take some energy in the form of GTP. When you're bringing things in, the GTP, this initiation factor is going to help this elongation factor is going to help make sure that it gets the right thing. It's all comfy and cozy in there. So this is going to help make sure that the anticodon matches the codon, at least those first 2 positions. And then you're going to have another elongation factor that's going to help push things over, because once this bond forms, then it's going to be awkward. And so you get another energy expenditure here too. But this actual bond formation, this isn't the part where we're spending energy, because we already spent the energy when we were charging charging the tRNA. And what we do is we make it so that we have this tRNA that is now going to be able to, in the help of the ribosome with this nice environment, perfect for mediating this attack where you have this amino group is going to attack, and so it's going to attack the carbonyl carbon where you have this attached to this tRNA, and so the new one is going to grab onto the growing chain. And the reason why it can do this is because you used energy to set up this system where you have these vulnerable bonds and you have things in the right position. And so the energy is going to come by putting this amino acid that you want to get added, by adding it onto the tRNA that has the matching anticodon. And so this is the job of the aminoacyl-tRNA synthetases, these aaRS's. And how they're going to do this is they're going to do it in 2 steps. And so it's going to be carried out by the same enzyme, the same aaRS, and we'll talk about this in a minute. And so the first step is going to be this amino acid activation. So it's activating because you're giving it this like high energy, you're attaching it to a high energy molecule, this AMP. So when we talk about high energy, we're talking about like potential energy. So I like to think of these ATP or GTP. The same thing happens with GTP. You just have a different base here. And so you remember the GTP and those other steps. But here we're going to be using ATP. And those other steps also, we're going from GTP to GDP. But in this step, we're actually going to be going from ATP to AMP. And originally this AMP is going to get stuck on the amino acid and then it'll get released. But so we call these high energy. You can think of these negatively charged phosphate groups when you're like pinching them close together. It's kind of like a stiff spring that you're clamping together. And so you can imagine that if you had a stiff spring and you put like a ping pong ball on the end, and then you let go, then it would go whew. But instead of going whew, what we're going to have here is the energy that would make the ball go whew, is actually going to be used to help get this new bond to form, and have things be all happy. And yeah, so basically this bond has high potential energy because of the whole like conservation of energy thing. If you takes energy to make it, you get back energy when you break it, and so we'll get back energy that we can use. And so, but so here in this example, we're going from ATP to ADP. And so then you still have another high energy bond because you still have those phosphate groups right next to each other that are vulnerable. But what we're going to be doing is we're going to be going to AMP. And so this is why I was mentioning earlier about how it's kind of like you have the two equivalents. And we'll get more into this in a minute. So our end goal is to get this AMP onto here. And so what we're going to have to do is we're going to actually have this is going to nucleophilically attack this phosphate. And so a nucleophile is something that has more electrons than it wants. An electrophile has fewer electrons than it wants. And these phosphates in these backbone, these phosphates are going to be electrophilic. They have fewer electrons than they want. Even though they're negatively charged, these groups are negatively charged, the oxygen is harboring all of the electrons. So the oxygens are pulling the electron density away from this phosphorus. And so it's going to be vulnerable to attack. And so it can be attacked by the nucleophilic carboxyl group. And so here it is, I have it shown in the carboxylic acid form, but typically at physiological conditions, this is going to be deprotonated. And this is going to give you a good nucleophile that you can then have attack. When you have this attack, then this will kick off. You get your pyrophosphate, and this can then further break down, hydrolyze into 2 molecules of orthophosphate, so individual phosphate groups. Okay, so now you have this. So why do we do this? Okay, so our real end goal is to get this amino acid onto a tRNA. So we're going to kind of like see this oxygen here. This part is going to get replaced by the oxygen of the tRNA. So we're going to be attacking the carbonyl carbon. We're going to be adding on to this carbonyl carbon. So right now, this oxygen, this is going to be where we're going to attack, and this is going to be kicked off. So if we look at it over here, we have this, remember, this is going to be like resonance stabilized in carboxylate form. You're gonna have this resonance stabilized. This is not, and even if it weren't, an OH is not a very good leaving group. And so what I mean by leaving group is that when we have a substitution reaction, so something is going to, the nucleophile attacks and then attacks the electrophile, and then it kicks off the leaving group. Because once you've attacked, now you have too many bonds and stuff, and so something's got to go, and so the leaving group has got to go. This reaction is not going to happen if that leaving group is not going to want to go, and so we want to make the leaving group want to go. An OH group is not going to want to go, and so but this AMP, this is a much better leaving group.
So this is a better leaving group. This is a more vulnerable bond. You've got this oxygen pulling things away, all of this group pulling things away from this carbonyl carbon, making this even more nucleophilic and now subject to attack by the tRNA, especially in the environment of the aminoacyl-tRNA synthetase. It's going to set up a nice environment where everything's properly situated, everything's right in place to react, as long as everything is the right size and that sort of thing. And so then you get this tRNA charging step where you take that adenylated amino acid. So it's got this adenosine monophosphate added onto it. And now we're going to kick that adenosine monophosphate off of it. And so here the tRNA is going to attack. You get the AMP off, and now you've got your aminoacyl-tRNA. And now you can spend ATP to regenerate this into ADP, which then you can use in the electron transport chain to make ATP again. So we've just invested all of this energy, and hopefully we got the right amino acid and the right tRNA. And so I'll tell you more about how this proofreading is done. Some of them have the direct, they can cut it off if it's wrong, but other ones recognize it well enough, hopefully, that you can recognize the right amino acid and the right tRNA, so that by the time this gets to the ribosome, you have the correct, that anticodon is corresponding to the correct amino acid. So all the ribosome has to do is look at this anticodon and see if it matches. And if it matches, it's going to fit nice and snugly in the ribosome. If it doesn't, it's going to be awkward, and it's going to push into a, it's not going to stay. And the GTP that it comes with, the initial elongation factor, is going to make sure that it is going to be the right one. And then it'll split ATP and leave, and then you can get the other elongation factor to push things over. We're like energy intensive, we're spending energy money, we're spending this ATP, and even worse, we're making AMP instead of ADP. So we're gonna have to spend an ATP to make ADP to make ATP, or ADP to make ATP. So we're gonna have to basically spend two more ATPs in order to regenerate ATP after this. So it's kind of like we spent 2. That's why sometimes you might see that things say like 4 instead of 3 per peptide bond formation. So we have the one where you have you're activating the amino acid and charging the tRNA. So this is going to take 1 ATP. Then in order to get that to the ribosome, you're going to have to spend a GTP. And then the GTP, it's going to make sure that everything is okay. Then it's going to get split. So now you've spent a GTP, and now things are going to you're going to spend another GTP in order to shift things over in the ribosome so that the A side is open once again. So that's where you get the 3 high energy bonds being broken, or you can say 4 if you're counting the AMP or the yeah. And I'll talk more about how this comes into play in energy in systems where, like in vitro translation systems, like this pure express system, which is used reconstituted from like purified components of E. coli. And they have like this energy regeneration system. And I found like a cool article talking about some of the things about energy requirements in vitro systems and what are some of the holdups. But first, I want to tell you more about these aminoacyl-tRNA synthetases, because we're spending this energy money. What we want to make sure, like, what is it giving us? So with this energy spending, we're going to be allowing this amino acid to be ready to be added to the protein, because this is going to give us the anchor so that we have this anticodon that is going to match to the codon in the mRNA, and you're going to have it held in the right position for this to then be attack the growing chain. And so imagine you have all these amino acids just like flying around, diluted all around the cell. it's going to be hard for them to find where they need to be, and how the heck is the ribosome going to know? But by putting them onto this tRNA, now you can have that happen. But because you're forming this other bond, you're decreasing entropy and all of this stuff, you're going to spend the money in order to do this. And by spending it in this enzyme, you're going to get the benefit of the enzyme's activity to make sure that the right amino acid is going to be added to the right tRNA. And so how is it doing this? So I found this really great review article. It's open access, so feel free to read, and I'll put the link describing these tRNA synthetases. So most of our amino acids, we have a single aminoacyl-tRNA synthetase that handles like all of the ones for that amino acid. So even though if we go back to our codon table, we have that redundancy where we have multiple codons spelling for the same amino acid. For most of our amino acids, a single aaRS is going to be able to recognize all of the codons for that amino acid. So like all of the tRNAs for that amino acid, which means recognizing tRNAs with multiple anticodons. Sometimes, like for some of these, they have to recognize like up to 6 different tRNAs. And so there are going to be different ways in which the aminoacyl-tRNA synthetase is going to recognize the tRNA. In addition to that, the base, so in addition to the anticodon part, it's also going to recognize other parts of the tRNA. And this is going to help allow it to recognize that tRNA. So it's not just the anticodon part. There's also different like identity elements that are regions of this tRNA that have either nucleotides, so either RNA letters, or structural things that are going to be recognized by the aminoacyl-tRNA synthetase. There are also different things that will make it so that it gets rejected by the tRNA synthetase. So that if it binds the wrong thing, there's going to be like anti-determinants that would similarly be like structural elements or particular bases, RNA bases, that are going to like clash with part of the tRNA synthetase, and then that's going to release the wrong one. So we call these wrong ones like non-cognate, and then the right ones would be the cognate amino acids. So there are like 23 known aaRS's, and so but there are only 20 common amino acids. So one of them, pyrrolysine, is only found in some little microbes, and I have a post on it, but it has its own synthetase, so that gives you 21. But what about those? We still have 2 to account for. There's also phosphocerol, which is another one that's restricted to some like bacteria and archaea. But then we still have that one, and this one we actually do have there's 2 lysine tRNA aminoacyl synthetases. So pretty cool. There are also different tRNA aminoacyl synthetases that our mitochondrial ribosomes use, but those are coded for not by the mitochondrial DNA, but instead by the cytoplasmic DNA. So they're made out in the nuclear DNA. So they're made in the nucleus, and then they get transported into the mitochondria. Okay, but so some of them have different weird mechanisms that they use. You can classify these aminoacyl-tRNA synthetases into like these 2 major classes. I'm not going to go into it. Some organisms have like more or less, so some organisms have like fewer of these aminoacyl synthetases, and some are have like really, really bad proofreading ability. I'll get to that somewhere in this paper. I remember reading it. It was pretty funny. Because we're talking back on the proofreading, we talked about how you recognize the tRNA, but you also need to recognize the amino acid. And so the amino acid, so there's going to be less things on the amino acid that you can recognize, because with the tRNA, you have this whole big structure thing. With an amino acid, you have like one little thing. And it could be really, really little thing in the case of glycine, or a big thing in the case of like tryptophan. So one way that the tRNA amino acid synthetase can discriminate between amino acids is based on their size, and also based on other features, like their charge, where they have potentials for forming like hydrogen bonds with the RNA, those sort of things. And so you have these, basically there's a couple of sites. So in addition to having this initial like kind of size, shape, all that, you're cool, you can bind. That's not enough for most of these. For about half of them, they also have some sort of editing part of them, and some of them have a transactivating or transacting proofreading thing, so like an editing partner, but many of them have this editing capacity inside of them, and so they have the second site. And typically there's some different mechanisms, but often what happens is that once the tRNA binds, then it kind of shifts things a little, that are going to shift that amino acid into the editing site. And there you have more contacts and things fit, so you can see, okay, is it really the right one? If it's not, then you can cut it off and boot it out. And if it is, then you get to keep it and keep on going. So with these mechanisms, you're able to make sure, for the most part, that the right tRNA has the right amino acid. But that doesn't mean that all is necessarily good to go. So if you can imagine that the ribosome can actually mess up and put the right put the wrong tRNA, the wrong aminoacyl-tRNA, and then load that. And so you can still get errors in your protein making. And so it is thought that there are like a miscoding event likely happens like every 1,000 to 10,000 codons. And so there's a lot lower fidelity, so a lot less exactly rightness of the protein making of translation than there is in like transcription, or especially replication. So DNA polymerases, when we're copying our DNA for like our permanent genetic instructions, that is going to have a super duper high fidelity. I'm trying to find the here. Oh, so DNA polymerase has like 1 times per 10 to the minus 8, because that's like our permanent stuff. So when we're making copies of our DNA, we want it to make sure that we're not passing down any mutations to future cells. RNA polymerase, so here we're making like our messenger RNAs as well as like our rRNAs, so like the ribosomal RNAs, various functional RNAs. Here you still have a great fidelity rate, but you're still making mistakes. Now you're making mistakes every like 1 in 10 to the minus 5th. But when you have the with the protein making, now you're having estimates that you have like 1 every 1,000 or 10,000 sites, you're going to have something wrong. And so basically 15% of average length proteins will have an amino acid mismatch. And so I actually didn't realize this before, so I thought that was a pretty interesting stat. But of course, these proteins are temporary. So one of the reasons why you can think like, okay, permanent, you need the most fidelity. Not permanent, but long-lived, you can make lots and lots of proteins off of an RNA. So if a messenger RNA has a mutation, then you're going to get lots and lots of mutated proteins. But if you make a single mutated protein during the process of translation, then that's like the least big of a deal out of the 3 of these. But when you have under various stresses and that sort of thing, the incorporation might get worse. And it can really be an issue. There are various mutations that people have, like genetic mutations, that can cause more misincorporation than usual. And there are actually some mutations associated with these tRNA aminotransferases, or aminoacyl-tRNA synthetases, such as this, like there's been mutations in various cancers, as well as I think I read like Charcot-Marie-Tooth disease has genetic mutations have been found that were associated with several of these transferases. So, and then there are some that are found in like mitochondrial tRNAs, synthetases that can cause various mitochondrial diseases. And so there are also these synthetases are also targets. So you can imagine that if you want to inhibit the activity of an organism, like a little bacteria or parasite, that sort of thing, tRNA synthetases can be a target for that. And some of these are actually known. So there's bacteria that make antibiotics for other bacteria to make sure that they don't infect them, or other parasites, or like fungi and that sort of thing. And then there are also synthetic ones that you can make. So one of these is, there's like this muropycin. This is a broad spectrum antibiotic. And basically what it does is it mimics isoleucyl adenylate. So it looks like it is an activated isoleucine, but it's not cleavable. And so it's going to get like stuck on. And then there are various other ones, puripermycin, basically, and there are ones that target different aminoacyl tRNA synthetases, and more in the works. But so the aminoacyl tRNA synthetases, though, that's just one step of the proofreading. And so that's just one step. But it is an important step because the ribosome, there you have a little more, you have that codon anticodon, but sometimes that can still get not happen properly, especially if the ribosome's like in a hurry, low on resources and that sort of thing. There was one I wanted to, sorry, there was this example that I was like super funny. There's some amino acid, some organisms that have like very little or no proofreading ability. And there's this one that they were calling, they had this really funny name for it. Oh yeah, statistical proteome, because the bacteria, these little bacteria possess highly reduced genomes that are either host restricted or intracellular parasites, but they have basically no mutated or degenerated editing sites in their synthetases. So they can make lots and lots of errors. And so this is kind of, I guess you have a lot of selection of the fittest happening in these. Just, I guess, hopefully good mutations are going to help them survive. Although, of course, here we're not passing down these mutations because this is just like randomly. So I don't know how they can be like randomly making proteins for the most part. But I don't know, maybe they're good with fairly good without the editing step. There are also other things. Other things that these aminoacyl tRNA synthetases have been found to do, especially in recent years, in addition to their roles in translation. So sometimes they could recognize things that look like tRNA and do things like, so there's actually this cool example in E. coli where they regulate transcription by binding to these stem loops. So these little like folded up stuff that mimics the tRNA, the tRNA for threonine, its anticodon arm. And so it's going to bind in front and activate or repress the transcription of the tRNA that it's made, like it's going to represses the transcription of more copies of itself. And there are also some other functions of tRNA synthetases, and it's a growing field of research. A couple more things. So sometimes, in bacteria, these are usually like standalone. They act by themselves, these aaRS's. In eukarya, so in like plants, animals, fungi, they often work in as part of complexes. And so this is the mammalian multi-synthetase complex. So you can see that you have like 9 different tRNA synthetases all working as part of this one complex. There was a couple other things I wanted to say. So the tRNA availability is actually going to be like one of the ways in which your cells are going to know how much amino acids are present. And if there are unacylated tRNAs, so there's not enough amino acids to be charging them, then these can actually bind to the ribosome and cause like signals to be given off in some bacteria. There's somewhere in here that I read it. I just don't remember exactly where it was, but I encourage you to read this paper. It's really interesting. But sorry, I'm just kind of like rambling and all over the place because I read this paper earlier today, and then I was like, oh, this is so cool, I wanted to say this, and then I was so cool, I want to say this, and then I really, yeah. But the last part is basically expand the idea of expanding the genetic code and using like artificial aminoacyl-tRNA synthetases. So even though we only use those like 20 amino acids, it doesn't mean we can't use others. We just don't have the machinery to make other ones. And so the machinery to make new tRNA aminoacyl-tRNAs is this aminoacyl-tRNA synthetase. But as I talked about, these have all these specificity things that to make sure that you don't add the wrong one. So now people are trying to engineer mutations into aminoacyl-tRNA synthetases to make synthetic ones that'll be better at adding on tricking. So sometimes you can like trick the ones into using them, but you can engineer them to be able to use even weirder amino acids. But then you have to have some sort of system where you've either removed the natural ones, so you're doing some like in vitro system, or you have somehow modified the genetic code. There's various things that you'd have to do, because then you have the whole codon issue of if you mutate the aminoacyl-tRNA synthetase, okay, well, what codon are you going to have it use, and various things like that. But cool stuff. There are also these really cool alternatives to aminoacyl-tRNA synthetases called flexizymes, and these are like they're RNA, they're ribozymes, so they're RNA enzymes. And they have these ability to add non-canonical amino acids on when they have these like activating groups, so they can like chemically design these amino acids to be added with all sorts of different things on them. And then the tRNA, it recognized, or this flexizyme, it's going to like recognize just the end of the tRNA. So it doesn't care about all the other stuff going on like normally aminoacyl-tRNA synthetases would care about. Instead, it's just going to care about the end. And so this is going to allow you to add on to basically any tRNA if you change the end of it, and then you can have different attacking groups and various modifications in order to get it to do the one that you want, the thing that you want. So yeah, something I only heard about recently, but apparently it's not that new. But yeah, I want to look into it more because just for fun, because it's really cool looking. Okay, but anyway, yeah, these aren't the ones that we have in our body. For these, you have to use them in like an in vitro translation system where you've kind of reprogrammed it. So you're taking out the stuff that would normally get the amino acid that would normally get charged on it and sticking this in. So you're kind of like replacing one. But I'm guessing that there are probably also versions where you can like add things. I don't know, but yeah, something to look into. Okay, speaking of in vitro translation, I found this cool article when I was looking into energy costs and that sort of thing. And so basically it was talking about how why energy is one of the big hurdles with in vitro translation is the enormous energy cost. And so in at least in these early systems, like only more than like 5% of the energy in these in vitro systems was actually being used for protein making, and the rest was being like uncontrolled and useless energy drains. And so, but in bacteria, like 70% of the energy consumption can be channeled into protein making. And so they point out that there's these three main issues. So one is this like metabolic energy drain I'll get into. One is that you have idle ribosomes and an excess of this elongation factor. And the third is the metabolism of amino acids. Basically in the ATP, like regenerating systems that they use, they're generating a lot of like pyrophosphate and orthophosphate and stuff, which are going to bind to magnesium and then like chelate the magnesium. And so remove the magnesium, and then this can cause a problem. And so there's different ways that can be made to like add extra things so that you can then regenerate and more things and regenerate more energy. But the other thing I want to mention from this is that this like idea of having excess EFG and idle ribosomes. So EFG, so that's going to be one of those. This is going to be the translocation factor. So it's going to be when you have that bond form, then you need to push things over. EFG is going to be the one that helps do the push-in. And so, but what happens is that this EFG, it can interact with empty ribosomes, or and so this can trigger the EFT. So the EFG, so basically when it recognizes a specific conformation of the ribosome, it can bind and then it can hydrolyze. And that normally pushes things over, but it can do this even if the ribosome doesn't have things to push over. So basically you're spending GTP needlessly. And so these systems like have to make sure that you have the right amount, the right ratio. So like 2 to 3 EFG per 10 ribosomes. And then also some of the amino acids can like degrade, which would block the protein synthesis, or yeah, so then you can add in more amino acids and get a better yield. And so people have been working on making things even more energetically efficient, and various things like that. So thanks for letting me ramble. I hope you found that at least somewhat somewhat interesting. Sorry it was all over the place, but yeah, I was just reading these papers and I was like, oh, there's all this cool stuff. And so I just wanted to talk it out. Okay, so maybe in the future I'll make a better version of this, but for now, this is just a talking out. But hopefully it helped.